Good afternoon, everyone. This is Kay from GenScript, and it is my honor to introduce our closing keynote speaker, Dr. Possu Huang. Dr. Huang received his Ph.D. from Caltech with the first demonstrations of a computationally designed novel protein-protein interface. He subsequently conducted postdoctoral research at the University of Washington before starting his group at Stanford University. He and his team focus on advancing protein engineering for novel therapeutics and protein-based nanotechnology. A couple of examples of their contributions include unlocking the design principles behind team spiral folding and their development of an engineered outer domain HIV proteins which serve as an improved immunogens, the latter of which resulted in a patent playing a critical role for vaccine development. His group uses machine learning, computational modeling, structural biology, and experimental libraries optimizations to continue the expansions of protein-based molecular platforms. His talk today is TRACeR, Antigen-centric MHC recognitions by a novel engineered protein platform. Dr. Huang, we're very thrilled to have you here with us. Welcome, and we look forward to the talk.
Thank you for the introduction. Yeah, it's very great to be able to tell people about the new research that we've been conducting. So today I'm going to talk to you about this new platform called TRACeR. Essentially, as the title suggests, that we now develop this new platform for antigen-centric MHC recognition. So just to start, I'll give you a little bit of a background for this motivation. Essentially, as probably everybody knows, that there are issues with targeting cancer. And this is a slide that I borrowed from this recent review that shows some of those biomarkers that are in development in clinical trials. As you can see, that the number of these markers are actually quite limited. And compared to the number of different tumor types, this is just a very, very small subset of markers that you can potentially target. And of course, the holy grail for this type of therapeutic approach is to potentially coming up with a strategy where you could actually access internal antigens. So all these, in the previous slide, all the markers that were being developed are mainly shown on the left side of this slide. There are generally either secreted proteins or cell surface antigens, and you can use perhaps a single chain FE or some other strategy to detect them from the outside of the cell. But when a cell undergoes transformation, or maybe perhaps being attacked by viruses, they will start to produce proteins that are anomalous to the cell, and then through this machinery. This is a general cellular machinery. Essentially, all proteins are expressed in the cell, would get chopped up and then actually be tested on these MHC molecules. And these MHC molecules will present a snippet that binds to them tightly to the surface of the cell. And this is a critical element for immunosurveillance. And this essentially is a way for cells to present the peptidome to the outside, to the immune system, so that potentially you could eliminate foreign or sick cells from the body when you actually detect these foreign antigen or disease antigen. So, and of course, these type of strategy is very useful for, say, cancer or viral infection. And of course, the MHC molecules are also responsible for a different type of disease, even though the processing for these antigens are a little bit different. But in general, your immune system takes this type of processed peptide presenting on the surface of the cell, and then that's how you interact with the immune system. But as I mentioned, the holy grail for this for immune surveillance, sort of like an immune response that model after this immunosurveillance, is to go through MHC. And in your body, essentially MHCs are these critical pivotal molecules that interact with the T cell receptor. So on this slide shows how natural immune system recognizes these MHC. So the TCRs shown here in blue essentially are molecules that looks like antibodies, and they also have 6 combinatorial loops that can actually recognize the antigen. So the footprint for these 6 loops, as you can see on the picture on the right-hand side, essentially uses 4 loops to anchor itself onto MHC, and then use the 2 more diverse CDR3s to recognize the antigen. So these molecules that recognize MHC is very exquisite in a sense that they've gone through this thymic maturation process so that the affinity is just tuned towards non-cross-reactive with the cell proteins. But there are actually very, very intriguing features that's associated with these TCR binding, right? Because there are only 1 trillion or approximately 1 trillion T cells, but there are more different type of scenarios that they have to come up with a response to. So the mechanism people have now proposed to be coupled to perhaps the mechanical coupling of these receptor to this T cell machinery, and that actually makes these TCRs very, very unique in a sense. So part of the feature that actually cells leverage is also by having this finely tuned affinity with the mechanical coupling, then now you can actually have binders are very, very weak in affinity, but then somehow you can still trigger very specific T cell responses. So these are very unique features that are associated with TCR, and they're just fascinating subjects to study. But generally, these are the only type of protein that we know can specifically recognize antigens by reading the peptide on the MHC. And of course, this implies that this type of molecules are very useful for immunotherapy, right? So now with the development of CAR T and then various bispecific antibodies, we now actually have a pretty good handle on how to activate T cells. But then, of course, the biggest question is how to actually find the target and only activate these specifically. So in this slide, specifically just showing you some uses for this type of TCR-based therapeutics. So traditionally, people use a single chain FV to activate CAR T cells, but now there are also these T cells are decorated with TCRs. They're called TCRTs, and then also there are commercial companies that use soluble TCR that were engineered to improve their affinity and solubility as a mono agent to actually activate T cells using soluble proteins. And also these T cell receptors are also programmed onto NK cells, for example, and they can also function like a CAR T type of behavior. So to use this TCR as a targeting molecule is not new, but the main hurdle is still coming back to this cross-reactivity, right? As I mentioned, these T cell receptors are evolved to be promiscuous because you only have 1 trillion of them, but they somehow has to recognize multiple antigens. And this is one of in the slide here. It's a very famous cross-reactive case where a T cell receptor can recognize 2 different antigens in practically the same fashion. So on the slide here, there's a known case for MHC, which is a tumor-specific target. And then in the magenta sense is that the target is called a fragment of titin. And as you can see from the A panel, these 2 antigens essentially are loaded onto the MHC in pretty much the same conformation. And the B panel shows that the T cell receptors that can recognize them is essentially bound to the MHC the exact same way. So this is a very well-known case for cross-reactivity, and this is causing serious concerns with this type of MHC-based targeting approach. And of course, to make matters worse, engineering these type of receptors. So first of all, like I said, natural TCRs generally has very weak binding affinity because you want to make sure that you don't cross-react at a high affinity. And at the same time, to actually achieve the specificity, of course, there are ways to do it, and you can do direct evolution to actually improve the specificity and KD at the same time. Alternative methods, including antibody-based TCR mimetics, haven't been pursued to try to replace this, or actually in parallel to supplement this type of TCR-based or TCR-like MHC recognition targeting capability. So in this picture, just to show you like some things that people have found in recent years. Again, the picture is just to show you that how these 6 loops engage the MHC and the antigenic peptide. And on the right-hand side from this paper, they analyze the footprint for TCR. So the TCRs are the blue molecules, and then there are antibody-based mimetics. They are shown in these orange structures. And when you analyze the footprint, it turns out that the different TCRs, of course, bind to the target slightly differently, and then they can pivot. But the CDR loops are still focused center around the antigenic peptide. But when you use experimental methods to develop these antibody TCR-like molecules, if you have any dominant epitope that could potentially take over the binding affinity, you would develop these antibodies that are binding off center, and then potentially most of your affinities coming from interacting with the scaffold instead of the antigen. So this potentially could also cause a problem later on for the developability for these type of strategies. So having telling you all about these type of concerns that is associated with targeting MHC, but still these MHC molecules are the sort of like the only way to actually glean into the cells to see the health of the state of the cell. And we really want to see if we can reimagine what a new solution might look like. So in here, the representation is that now we have a cell surface, and then now you have a lot of MHC molecules display on the surface with different antigens. And of course, the ideal scenario is to come up with a platform very much like how the TCRs would behave, where you would have an element. Now, you need to make sure that this binder can recognize MHC specifically, right? Because you don't want to have a new protein or some other molecule that just go around and bind to any surface protein. So it has to have specificity for MHC class 1 or class 2. But the critical juncture here is to actually develop ways so that you can recognize these antigenic peptide. So, and of course, as I've shown you with the TCR examples before, that these 6 loops from the TCR and extendable to antibodies just sets it up so exquisitely that the antigen recognition comes very naturally. Of course, evolution provided these examples. But in order to actually improve the engineering capability for us to target these MHC molecules, we wonder if we can actually go back to the drawing board and then come up with a new design that actually has a similar feature, but actually would have these new features that are much easier to engineer for one, and potentially you will actually make a single chain protein that is more stable and easier to produce for implementation. And of course, since these are proteins, the ends are usually free, so you want to have a scenario where you can actually be modular and be compatible with any functional payloads. That could potentially be T-cell activating molecules, or it could just be decorating onto T cells as part of the CAR T, or you can just deliver them as drug conjugates. So it should just be a modular targeting component. So, and interestingly, we happened to come across this molecule that kind of gave us an inspiration for how to solve this problem. So this orange molecule in the middle is a superantigen. This structure has been known for about 15 years, and people actually haven't tinkered with it because they're actually somewhat difficult to deal with. So these superantigens are, as you might know from your immunology knowledge, that they generally cross-links TCR and MHC class 2s to activate CD4 plus T cells in a non-antigen specific fashion. So they are just molecular glues that can activate T cells. And generally, these superantigens just don't have the built-in features to recognize the antigenic peptide. But the inspiration we take from this molecule is that we now, in addition to TCR, we actually have a mode of attack to these MHC molecules, because now this molecule shows that you can simply use a helical bundle and just have it sit in sort of like saddling over these MHC molecules and allow this molecule to latch onto MHC, and potentially you can actually use that to leverage the structural feature for antigen binding. So, but as I mentioned, these superantigens, they generally interact with MHC class 2 in a non-antigenic specific fashion. So how do we actually turn that into targeting MHC1 for potentially cancer immunotherapy? And fortunately, as you probably already know, that these MHC1 and MHC2 shares general structure features. And the pictures shown here are just listing the subtle differences between the two. Generally, the binding pocket for the MHC1 just made out of a single chain, but the MHC2 is double chain, now shown in 2 different colors. And perhaps we can leverage this structural feature to develop our binder interface that now allow us to actually cross these binding features from MHC2 to bind MHC1 protein. So, and this process generally they are the MHC1 and MHC2, fortunately, again, provided by nature this example of TCRs, because even though there are 2 different types of MHC, class 1, class 2, but there's only one type of TCR. So TCR recognizes both classes. So that basically gave us a clue suggesting that this cross-reactivity, crossing over reactivity must be achievable. So essentially what our goal in using our new protein design knowledge is to potentially taking this MHC2 engaging pattern and try to develop something that looks like it, but can engage MHC1. And of course, this type of technology is only available in the last probably 5 to 10 years, and that wasn't available before. So probably that was the reason why nobody really tried to develop these superantigen-based modifications. So in terms of how we would go about developing this, I'm not going into the technological details because I won't have the time for doing this. But this slide generally shows the computational pipeline that we use. We essentially build these helical bundles, and we need to understand how it interact with these 2 helical structures that's on the MHC1, and how you could potentially use different orientations to approach the antigen that's embedded in the middle. So we use Rosetta as our modeling tool, and it turns out that we could actually use just the same saddling orientation to model these molecules. And by Rosetta calculation, we can actually see all these different sort of orientation relative to the MHC1 that we can actually engage these helical bundles and still have access to the peptide. So with this, it turns out that with the different docking modes, they give different computationally calculated energy signature. And on the right-hand side, just basically to illustrate that some orientation will give you a better computational energy for the interface. And it turns out that for the final binding mode that we chose, it actually looks very much like the MHC2 engager. So it looks like there are some very interesting shape that these MHC molecule presents to other proteins that we can leverage. And it seems to our modeling effort at least suggests that there's a convergence to latching onto these structural features. So with this modeling in hand, we then want to use experiments to actually test them. And this is just a very quick description of the type of target that we use for these experiments. We work with Nick Skorakis. His lab developed this very convenient peptide exchange method to actually produce MHC1 tetramers very, very quickly. And essentially, we can actually get get reagents from them within a week of like identifying the target. So we use their reagents for this experiment, and we carry out the experiment on yeast display. So you're going to see a lot of data pictures like this in the next few slides, and essentially these are yeasts transformed with our binding protein, and then they will be displayed on the surface of yeast. So you can see that these are cytometer readouts. So generally you see these two lobes, if you see the lower left-hand corner. These would be two populations. The one on the left are generally considered as yeasts that are not expressing or lost the plasmid, and the one on the right-hand side would express our protein on the surface. So if we have a binding event to these MHC molecules, soluble MHC molecules, you will see that the right-hand lobe would shift upwards. So, and essentially this just to tell you that we essentially take our design coming from the computer, we build a library, and this is using GenScript's precision library because now we have way too many positions to cover, and the diversity can be precisely controlled through this precision library. So we implement this library into the yeast platform, and we just use these soluble tetramers to screen. And the bottom panel showing these 5 different steps is just the enrichment rounds for these computationally generated focus library on yeast, and when we use these tetramers to screen them. And by the 4th round or 5th round, you can actually see this very distinct subpopulation that already show binding. So this is incredibly encouraging because then now we actually have binder. We can use a similar, we don't know if this is a similar binding mode, but at least we have designed it and modeled it this way, and hopefully that binds exactly the same as the way we model it. And what's more surprising is that when we isolate the best binder from the previous enrichment experiment, just by taking that single clone and test it against different MHC alleles, it's already very specific, and it's only specific to the allele and to the peptide. So the top row, the 3 of them, the one on the left is the positive control. So there's a single clone binding to its cognate target. The middle one is actually using an MHC2 tetramer, and the one on the right is actually the HLA-B, that is a different allele of the MHC1. So as you can see that there's no cross-reactivity for these different MHC alleles. But for the bottom row, we essentially take the same MHC, load them with different peptides, and you can see that it's actually peptide-centric because now our binder just does not cross-react even if you use the exact same MHC background. For this type of specific recognition, now you actually make our life very, very easy to develop this new platform, right? Because I just show you that, through this computational modeling way, we figure out that there are these helical bundles that we can design that can be very specific to MHC allele and then to the peptide. So we now build this scaffold of the bundle scaffold. We call that the TRACeR platform. And now essentially, since now we know that you can recognize the MHC specifically for the peptide through this element that we engineer, and all the recognition elements essentially just sit on this one loop. And again, this is using the precision library that is provided to us by GenScript. We can now just decorate this loop with like 19 amino acids at each position, so we can avoid the stop codon. And actually use yeast to very easily select for binders for different targets. And this slide shows you like how easily that could be achieved. So essentially we have a master library, and then now we actually throw at it 3 new targets. The top one is the same NY-ESO1 target, which is a tumor specific antigen. But then we tried it against EBV, who's the viral peptide, and SARS-CoV-2 on a different allele. And the lower right-hand corner shows you how each of the selected binders, so each row is a single clone tested against the 3 different targets. And you can see that only the diagonal has the binding signal, so it shows that we actually now have very, very specific binder for each of the targets, and you're all coming from this one single master library. That is also very easy to handle. So for the interest of time, I will fast for the next 2 slides, but actually these are very interesting observations that we now made, because with the interest of targeting tumor specifically, we actually took this NY-ESO1 antigen, and now this picture is showing you how this peptide is actually loaded onto the MHC. And so the text is a little bit messed up here, but essentially we label each of the position of the amino acid that's actually part of this antigen. And we color these 4 different positions in yellow, because these are the residues that are pointing upwards. So if you want to gain access to this antigen, you essentially, these are the 4 amino acids you potentially have access to. And what happened if we actually do alanine scan against this entire peptide? And it turns out that the binders is exquisitely specific. So as you can see on the bottom panel, now with this point mutation of these positions to alanine, for 3 of those upward facing positions, we lost all binding. And this is very, very significant, because now imagine if you change the first position from serine to alanine, the methionine at 4 and tryptophan at position 5, they are still there. But for some reason, all 3 residues are required for binding. And we're still trying to get the structure of this binding mechanism. But the way I can imagine it is that you now have 3 anchor, and essentially the binding loop acts like a rubber band, like sort of wrap around them has to be somewhat. And if you lose any one of those position, you would just be incomplete, and actually would just fall off. And one question you might raise is that what happened to the Q8, right? Like this far end position, somehow that doesn't affect binding. And I think that's just because the way we model these binders, QA is way too far from our binding footprint. So the binder doesn't interact with it, and that's why there's no sensitivity. Now that we actually discover that there are 3 positions that are responsible for our recognition, we want to really define the breadth of specificity for our binder for this NY-ESO1 target. And we essentially do site-saturated immunogenesis for each of the positions. And in magenta, now we show each of those interacting positions mutating to 19 other residues. And you can see, just even from the colored block, shows that our binding is very, very specific to NY-ESO1. So the top row, because we only see a little bit of cross-reactivity on the top row, so the top row, raw data for the top row is actually shown right below the table. And you can see this is what we mean by cross-reactivity. So naturally, this position is a serine, and when it's mutated to threonine, because it's isosteric and has the same chemistry, it's expected that you could potentially react with the threonine. But we interact very, very weakly with the cysteine in this row. And then all the other rows are essentially just silent, and there's no signal at all, so I'm not showing those. But interestingly, when we compare this profile to published results, so Molecular Partners posted their DARPin-based binder against also NY-ESO1 on their website, so we can actually see, compare our specificity to theirs. And as you can just kind of see where the colored block shows up, our binders are actually very, very specific to this particular target. So now that we actually have this new platform, we can make just do very simple selection for a very specific peptide. What we're doing now is actually developing this pipeline to make very fast turnaround and to engineer new targets. So this just shows you a little bit of a timeline of like the estimated timeline for developing any new binders. So the top one is just a selection process. We conservatively say that it takes a month, but really it only takes about 2 weeks. But once you do the mutational scan to identify what residues interact with and what amino acids could potentially cross-react with, what we do is to go to at least all the known antigenic databases to identify what could potentially cross-react. And if they cross-react, we can actually always go back to make negative selection probes to actually refine our binder. So this is now the pipeline that we're building to create these TRACeR binders. And of course, the idea is that perhaps we can use it, like as I mentioned before, directly as these modular targeting molecule, and it can be implemented in like any format that you want for therapeutic applications. So I'm just right on time, and just the last slide, I have to acknowledge my super talented graduate student that really pulled this off. And this entire project, all the data that I show here is done by Tian-Tu in my lab. And this project is supported by American Cancer Society, Stanford, and also we get a lot of help from GenScript creating these libraries. So we really appreciate those assistants. So with that, I will end here. Happy to take questions.
Hello, this is Kay again. And thank you, Dr. Huang, for sharing the very interesting work and finding. And we do see potentially this platform could be a game changer for cancer immunotherapy, and even a strategy that combines therapeutics and diagnostics, and also a way to engineer different specificity. So for audience who has questions for Dr. Huang, feel free to put it in the Q&A box here. We'll try to accommodate as many questions as we can here with our time. Let me pull some questions up. So the first questions I have here is on the potential application for this platform here. Have you ever extended this platform to different MHC alleles?
Yeah, so we, as I showed you, we actually were able to actually show that we're very specific to one allele, and we can actually potentially cross-react with the HLA-B. And actually now we have, but we have not tried to engage HLA-B. But we do actually have preliminary data suggesting that we can actually access MR1 as well. So for those of you who may not know, the MR1 are these molecules that actually load metabolites instead of peptides. And it turns out that our platform might actually be able to target those as well.
Thank you for that. And do you mind expand a little bit on the functions or importance of MR1 from there on the? Yeah.
So the function. So essentially, people. I think there's a paper in like the last 3 years. People have identified that now MR1, even though it's picking up metabolites, but upon transformation into malignant tumor, the metabolome changes. So actually the metabolites going through MR1 and being presented on the surface of the cell change according to that. And so there is actually a correlation with whatever changes inside of the cell could directly change the display pattern of MR1. And they have found that if you can target MR1, you could potentially actually kill a large different type of tumors. So it's very interesting, but it's very, very preliminary. We didn't actually have a lot of data on that yet, but the preliminary data definitely shows that our strategy works on MR1.
Thank you. Very, very interesting, and hopefully we'll see more work on that. The next questions is on, I think, the tools Rosetta for the screening on the algorithm. Could you expand a little bit, and is it mainly based on protein structure, or is it using experimental data feeding and learn and predict?
Yeah, no, this is all built by hand, actually. So the oh yeah, now I actually see the tab with the Q&A. The protein prediction tools cannot predict this molecule, because the interesting thing that we did was to take this helical bundle, but then as you see, the uniqueness is that there is actually a loop stretch over the surface of it, and that part seems to be novel, and most predicting software couldn't actually predict it correctly. And then it's mainly based on and the experimental data to feedback for a learning cycle. We were actually thinking about doing that because our binding footprint, the sequence that goes into the binding footprint, is actually very, very small. As I mentioned, it's just literally a few positions we can just saturate it. And if we accumulated enough correlation between these sequences to the target antigenic peptide, we could potentially just build a machine learning tool to actually generate new ones for new targets. But we don't exactly know how far that would go yet. But that's definitely something we'll consider. But in order to actually get that data set, I think it will require a lot of binders processed through this platform. So we don't currently have that super.
Got it. Thanks for explaining. On the binder side, we also have people asking about whether you have data on the range of binding affinities for these binders.
Yeah, so currently we mainly focus on the specificity because I think that was the biggest challenge to solve. But so these binders, the ones I show you, they bind in a wide range of affinities. The tightest ones we get are around 50 nanomolar, and then they go all the way to like 500 nanomolar. So it's like somewhat of a low mid nanomolar range, and we are now also pursuing optimization to see if we can actually improve upon it, because I think our initial platform was just to try to see if it works, and then now we're actually spending time on optimizing it. I think the KD would definitely be improved in the next iteration.
Thank you for that. I think with the interest of time, I'm going to take the last questions here. It's again, this question's on the platform itself, whether you see this platform be able to differentiate antigens MAGEA3 and Titin. I think it's peptide derived from a cardiomyocyte protein.
Right. So that was the example I showed you earlier, saying that this is like a known challenging problem. So we actually already have binders for those two, and because the way the antigen is loaded, our binder actually also binds to both of them. And we just got a new set of MHCs that are labeled with a different color, and now it enables us to do the negative selection. And we're actually in the process of figuring out whether we can actually differentiate the two. But as of right now, we have binders for both of those, but we haven't actually see if we can separate them out yet.
Got it. Thank you for that. With the interest of time, we'll stop the Q&A here. But again, thank you very much, Dr. Huang, for the excellent talk here. And we're very happy to have you. For the audience, this is our last talk. But stay tuned, don't leave yet. We will announce the winner for the scavenger hunt within 10 minutes. So stay tuned. And our next session is on the closing talk, will be around 5:35. Again, thank you very much all for joining the talk today, and have a great day.